Ex-Situ Conservation. Ex-Situ Conservation means the conservation of species outside their natural habitats. Ex-Situ Conservation means the conservation of species outside their natural habitats. It involves the transfer of genetic material away from the location where it is found. It ensures easy accessibility of germplasm for evaluation and utilization. Simply in one word, ex-situ conservation means conservation of species outside their natural habitats. Ex-situ conservation, the protection of endangered and genetically modified species away from their natural habitats under human supervision is known as ex-situ conservation. In this ex-situ conservation, mainly we focus on endangered species like plants and animals of endangered and the genetically modified plants and genetically modified animals away from their natural habitats under human supervision is known as ex-situ conservation. Ex-situ conservation is the best way to instantly protected rare and endangered species. So instant result is done with ex-situ conservation. Spontaneous results done with ex-situ conservation. Plant and animal species are at risk of extinction due to the factors such as inbreeding, genetic drift, climate change, habitat changes. So it is being artificially preserved through foreign care. Some types of ex-situ conservation methods here it is. One is genetic reserve centers, germplasm banks, gene banks, cryopreservation, botanical gardens, zoo parks, seed banks, pollen banks, aquariums, NBPGR, NBAGR, CIMAP, ICRISAT. These are the methods of ex-situ conservations. So genetic reserve centers, germplasm bank, gene banks. These three types of ex-situ conservation methods for storage of genetic material. Complete genetic material is stored in their artificial environment, in their laboratories, in the cryopreserve centers. So plant and animal genetic material or germplasm is stored in the ex-situ conservation for future purpose. Cryopreservation, the most advanced technology that preserves living parts for a long time. This type of care involves plant and animal parts with the help of dry ice in liquid nitrogen at a very low temperature at minus 196 degrees Celsius. In this long-term care, at least 12 years at very low temperatures, the metabolism of organisms almost freeze. So at the low temperature, the storage parts of living organisms, like maybe plants and animals. The total metabolic activities are infreezing, so they are cannot involved in the metabolism activities. It is a advanced technology for the storage of genetic material, living organism spots for a long time. The time is at least 12 years. Next one, ex-situ conservation method is seed banks. Seed banks are where seeds are collected and stored under favorable conditions for the future uses. Seeds can be divided into two types. They are orthodox seeds. Orthodox seeds that have been stored for a long time without genetic degradation are collected, called orthodox seeds. Seeds can be stored for a long time by maintaining high temperature and low humidity. The second type of seeds recalcitrate seeds. Seeds that cannot be stored in seed banks are called recalcitrate seeds. They do not survive low temperatures and dehydration. Example, pineapple, citrus, coffee, tea, and mango. Zoo parks. It is another type of ex-situ conservation method of animals. A zoo is a facility in which animals are housed within enclosures cared for, displayed to the public, and in some cases bred for conservation purposes. Botanical gardens. It is one type of ex situ conservation method for the plants. Royal Botanical Garden preserves the British flora of 1500 species. The garden is full of flowers, nuts, and fruits. The gardens create a peaceful and beautiful atmosphere. The Royal Botanical Garden alone preserves 2,500 endangered species worldwide. And next type of ex-situ conservation method is pollen banks. The pollen is used directly in methods such as food, plant reproduction, and artificial pollination. So, strong pollen is very necessary for the future generations. They are used for the food material, they are used for the plant reproduction and artificial pollination. The pollen banks, mainly done with cryopreservation, is the process of preserving in liquid nitrogen with dry ice at minus 196 degrees Celsius. Example, mango, citrus, tomato seed, pollen grains. And CIMAP, Central Institute of Medicinal and Aromatic Plants. The research center is in Hyderabad. It is also one type of ex situ conservation center. This center mainly focus on medicinal plants in India, not only medicinal plants, including aromatic plants. The cultivation, preservation of medicinal and aromatic plants in the center of Central Institute of Medicinal and Aromatic Plants. It is a research center. It is located in Hyderabad. And ICRISAT, International Crop Research Institute for Semi-Arid Tropics. The center is located in Hyderabad. At Hyderabad, the ICRISAT Center mainly focus on semi-arid tropic areas. in semi-arid tropic areas having which type of soil content, and in this soil content which type of plants are more gives yield to the peoples. So the center is research on this semi-arid tropic areas and semi-arid tropical plants. These are the ex situ conservation methods for the biodiversity.